All right, buckle up, everybody, because today we're going deep into stem cells.
Deep, deep.
But not just like the stem cells 101 stuff.
Right.
You want to know about the real puppet masters.
The puppet masters, yeah.
Those tiny proteins, the transcription factors,
Yeah.
that are actually controlling a cell's destiny.
That's right.
These are like the conductors of the cellular orchestra,
They are.
deciding, which genes get played and ultimately shaping what a cell becomes.
Absolutely. So TFs, or transcription factors for short,
Okay.
they're proteins that bind to DNA.
Okay.
And they act like switches, turning genes on or off.
So essentially, it's like this massive control panel with thousands of switches.
Yeah, think of it that way.
And each one is like controlling a different...
Different aspect of the cell's identity.
Yeah. Wow.
Mm-hmm.
So, I mean, that sounds a lot more complicated than just an on and off switch.
Oh, it is. It is.
Yeah.
It's way more nuanced than that.
Okay.
It's not just about whether a gene is on or off. It's also about the level of activity. How much of a specific protein is made.
I see. So like a dimmer switch.
Yeah, like a dimmer switch. Exactly.
Not just on or off, but like how bright.
Exactly.
Okay.
Exactly.
Okay.
And these TFs, they don't work in isolation.
Oh, I'm sure.
They interact with each other,
Right.
forming these complex networks
Yes. Yeah.
to really fine-tune gene expression.
So it's almost like a circuit board then.
It is.
Yeah.
It is a very intricate circuit board.
Wow. And knowing how precise, biology is.
Right.
How in the world do these cells manage to direct stem cells towards specific fates?
Right. So let's start with embryonic stem cells.
Okay.
The ultimate source of
Oh,
potential.
the blank slate.
Yeah, exactly. The blank slate. And there are three really, really important TFs there that you got to know.
Okay, lay it on me.
OCT4, SOX2, and NANOG.
Okay.
These guys are essential for maintaining pluripotency. Pluripotency, which is the cell's ability to become any cell type.
Right.
So they're essentially the guardians of stemness.
They're holding it.
They're holding it in that state of, I can become anything.
Pure potential.
Pure potential.
Yeah.
And it's not just about having them there.
Okay.
Like take OCP4, for example.
Yeah.
It's incredibly sensitive to its own levels.
Oh, really?
Yeah. Too much OCT4.
Oh.
And you lose pluripotency.
Interesting.
Too little, and you also lose pluripotency.
It's like the Goldilocks.
It's the Goldilocks protein.
It's got to be just right.
It has to be just right.
Wow, that's so interesting.
It's really a delicate balance.
Yeah, it is.
And it highlights how sensitive these systems are to even like small changes
Oh, for sure.
in concentration.
So, I mean, they can't stay blank slates forever, right?
Right.
They eventually have to actually pick a path.
They do.
So how does that transition happen?
Right. So that's called lineage specification.
Lineage specification.
Where a stem cell commits to a specific fate.
Got it.
And guess what? Transcription factors are the key players here.
Yeah.
Different TFs guide those cells towards specific lineages.
So, whether that's becoming a neuron or a muscle cell or a blood cell.
Exactly.
Each of those different types has their own set of TFs.
They do. So they needed
their own little team.
Yeah, their own little team that activate the genes needed for that cell type.
Okay.
But they also suppress the genes that would lead to other fates.
Interesting.
So it's really carefully controlled.
So it's like this dance, with different TFs kind of taking the lead at different points.
Perfect analogy.
Wow.
It's a beautifully choreographed dance.
Yeah.
And that kind of brings up the question.
Yeah.
How does a cell make sure that the right TFs are activated?
At the right time.
At the right time.
Yeah.
And what are the fail-safe mechanisms?
Yeah, like how do they not mess up?
Exactly, to prevent errors in this choreography.
What happens when they do mess up?
Well, that's when you can see developmental defects.
Okay.
Or even diseases arise.
Okay.
I see. Because if the wrong TFs are activated or deactivated, the wrong time, it can disrupt the whole developmental program.
So it's not just like which genes are on and off.
It's not.
But it's also the timing.
The timing.
And the precise levels of these TFs.
The precise levels, absolutely.
Yeah. That's incredible.
And to make it even more complex,
oh no,
there are other players in this orchestra.
Oh gosh, okay, like what?
So for example, you've got epigenetic modifiers.
Epigenetic modifiers, okay.
These can actually alter the structure of the DNA itself.
What?
Yeah.
Wow.
Making it easier or harder for certain genes to be accessed by the TFs.
So they're like setting the stage.
They're setting the stage.
To make sure that the conductor can reach the right genes.
Precisely.
Okay, wow.
And then you've got these non-coding RNAs
Non-coding RNAs.
Which can interact with the TFs or even directly with the DNA to fine-tune gene expression even further.
Oh my gosh, it's like this whole team of stagehands.
It is.
Working behind the scenes to make sure that this performance actually happens.
To make sure the performance goes off without a hitch.
This is incredibly complex.
It is.
How do researchers even begin to try to figure this out?
Well, it's definitely a challenge.
Yeah.
But there have been incredible breakthroughs.
I bet.
In recent years.
Yeah.
So, for example, CRISPR-Cas9 allows us to make very precise changes to genes.
Oh, wow.
So we can essentially manipulate the score of the orchestra.
So you can just like go in and
edit the sheet music and see how the music changes.
That's amazing.
And then techniques like single-cell RNA sequencing that allows us to analyze the gene expression of individual cells.
So we're getting so much more granular with this.
We're getting incredibly detailed information about which genes are being turned on or off.
Oh, wow.
In different cell types.
So we're understanding the language of cells now.
We are starting to understand their language.
And how these tiny proteins are orchestrating their fate.
Absolutely.
It's leading to some incredible medical breakthroughs, right?
Oh, absolutely.
Like with induced pluripotent stem cells.
Yeah.
And direct reprogramming.
Direct reprogramming, yeah.
That's amazing.
It's really incredible. We're learning to manipulate cell fate in ways we never thought possible.
I am blown away. This is incredible.
Yeah, it's a really exciting field.
Just scratching the surface here.
Yeah.
And already mind-blowing, but I think we need to take a pause.
Okay.
Process all this information, because when we come back, we're going to delve even deeper into the therapeutic potential of these transcription factors and explore how they're actually revolutionizing medicine.
Sounds good.
Stay tuned. Welcome back, everybody.
Welcome back.
You know, we've been talking about these fascinating proteins, these TFs, and how they're essentially controlling a cell's fate. It's amazing that we're starting to, like, understand the language of cells and manipulate it.
Yeah, we're actually...
It's incredible.
We're really starting to learn how to speak their language.
Wow.
And it's opening up some really incredible opportunities in medicine.
Yeah, let's talk about that. So before the break, we touched on induced pluripotent stem cells and direct reprogramming. Can you give us like a concrete example of how this might actually work in a medical setting?
Sure. So let's imagine that somebody has a spinal cord injury.
Okay.
You know, the nerve cells in the spinal cord, they don't regenerate very well on their own.
Right.
And that can lead to permanent paralysis.
Right.
But what if we could take cells from the patient's skin, reprogram them using specific TFs, and convert them into new nerve cells?
Wow.
That could then be transplanted into the damaged area.
So you're essentially like bypassing the body's limitations.
Yeah, exactly.
You're just creating the cell that you need.
You're creating the exact type of cell you need for repair.
Like a cellular repair kit.
It is. It's like a cellular repair kit.
And the TFs are the tools.
The TFs are the tools. That's right.
I mean, that sounds...
It's powerful stuff.
Almost too good to be true. So what are the challenges?
Sure.
You know, with making these therapies like a reality for people?
Well, safety is a big one.
Yeah, I can imagine.
You know, we need to make sure that the reprogrammed cells actually behave as intended and don't pose any risk to the patient.
Right.
So, for example, what if those reprogrammed cells start dividing uncontrollably?
Yeah.
That could lead to tumor formation.
Oh, right.
So we really need to make sure we understand.
So it's more than just like flipping the right switches.
It's more than just flipping the switches.
You've got to make sure the whole...
Yeah, the whole system needs to remain stable and predictable.
It's got to be controlled.
Exactly. We need to avoid any unintended consequences.
Right. So how do you even begin to do that?
Well, one way is to use very carefully selected combinations of TFs. And really fine-tune their delivery. And make sure those new cells integrate properly with the existing tissue.
So it's a balancing act.
It's a very delicate balancing act.
Yeah, because you're essentially like rewriting...
You're rewriting the identity of the cell.
Yeah.
So, yeah, precision is paramount.
What about, you know, the efficiency of this? Are we able to reprogram cells?
That's another area of active research.
Okay.
You know, we're still learning how to optimize that process to make it more efficient and reproducible.
Right.
And some cell types are easier to reprogram than others.
Okay.
And there's a lot of variation between individuals.
So it's like fine-tuning a recipe.
It is. It's like fine-tuning a recipe.
Yeah.
You know, experimenting with different ingredients and techniques.
Until you get it just right.
Until you get it just right. And that's where personalized medicine comes into play.
Oh, interesting.
Because as we gather more data about how these TFs work in different individuals and different cell types, we might be able to develop like customized strategies.
Oh, wow. So it's not like a one-size-fits-all.
It's not one-size-fits-all.
You can actually tailor it.
You can tailor it
to a specific person.
to each patient's unique needs.
That's incredible.
Yeah, imagine being able to like analyze a patient's cells.
Yeah.
Identify the TFs that are dysregulated in their disease.
Right.
And then develop a therapy just for them.
Wow.
That's really the future of precision medicine.
It's mind-blowing to think how far we've come. You know, we're not just observing cells anymore.
Yeah.
We're learning how to like communicate with them,
Right.
guide their fate.
Yeah, it's really incredible.
And we're just scratching the surface.
Yeah, and we're just scratching the surface of what's possible.
There's so much more to discover.
So much more to discover.
About these proteins and how they can revolutionize medicine.
Yeah, imagine being able to regenerate damaged heart tissue after a heart attack.
Wow.
Or repair a severed spinal cord. Or even, slow down the aging process.
It sounds like science fiction.
It does, but it's becoming our reality.
Are there any, areas of research that you're particularly excited about? Like, what's on the horizon?
Well, one area that I think holds immense promise is the development of cell-based therapies for neurodegenerative diseases like Alzheimer's and Parkinson's.
Oh, wow.
So these diseases, they're characterized by the loss of specific types of neurons in the brain.
Right.
What if we could generate those neurons from a patient's own cells?
Okay.
And transplant them back into the brain?
Oh, wow.
We might be able to slow down or even reverse the progression of these diseases.
So it's almost like a cellular tune-up?
It is. It's like giving the brain a tune-up.
Replacing those damaged parts.
Yeah, with fresh, healthy cells.
That'd be incredible.
Yeah, and it goes beyond replacing those cells.
Okay.
We're also exploring ways to use TFs to modulate the immune system.
Oh, wow.
Enhance tissue regeneration. Yeah. And even target cancer cells more effectively.
It sounds like we're really on the cusp of something, the whole new era of medicine.
We're moving into a new era of medicine.
Where we can, harness the power.
The power of our own cells.
Of our own cells to heal.
To heal and regenerate.
But with such incredible power, comes responsibility.
Absolutely.
What are some of the, ethical and societal considerations that we need to be thinking about as these technologies advance?
That's a really, really important point.
Yeah.
Because with any powerful technology, there's the potential for misuse.
Right.
And we really need to think carefully about the ethical implications.
Yeah.
And establish clear guidelines
for
the development and use of these therapies.
This is a fascinating topic.
It is.
So many different layers.
Lots of layers.
We're going to take a break.
Okay.
When we come back, we'll dive into those ethical and societal implications.
All right.
Explore the challenges and opportunities
That's good.
that lie ahead as we continue to unlock the secrets of cellular destiny.
Big questions.
Stay with us. Welcome back, everyone. We've been on quite a journey.
We have.
Exploring the world of stem cells and these powerful transcription factors, shaping their destiny. And it's incredible to think that we're starting to unlock these secrets and even, to some extent, rewrite it.
It is pretty amazing.
But as with any powerful technology, there's ethical and societal implications we really need to consider.
Oh, absolutely. We've talked about some of the potential benefits, right?
Right, yeah. Treating those spinal cord injuries.
Spinal cord injuries, regenerating organs.
Yeah.
Even tackling those neurodegenerative diseases.
But, as we delve deeper into manipulating these cells, it begs the question, where do we draw the line?
Yeah, where is that line?
The potential to improve human health is undeniable.
Absolutely.
But what about misuse?
Right.
We've all seen those sci-fi movies, right? Yeah.
Where the genetic engineering goes awry.
The classic trope.
Yeah, and it's easy to just dismiss those as fantasy.
Yeah.
But with the rapid advancements in this field, it's a conversation we need to have.
We do, for sure. And I think one concern that comes up a lot is this idea of designer babies.
Oh, right.
Using these techniques not to treat a disease, but to actually enhance traits.
Like intelligence or athletic ability.
Intelligence, athleticism, yeah.
Yeah.
And I think that brings up some serious ethical questions about what it means to human,
Right.
Should we be tinkering with the very building blocks of life?
It's a slippery slope.
It is. It is.
Like, where do we draw that line between treatment and enhancement?
Yeah, and who gets to decide?
Yeah.
That's a big one. It's not just scientists who need to be part of this conversation.
Right.
This is something that society as a whole needs to grapple with.
Absolutely. And it's not just individual choices either, right?
No, there's those broader societal implications.
Absolutely.
Imagine a world where these therapies are only available to the wealthy.
Right. That would just exacerbate existing inequalities.
Yeah.
We need to make sure that everyone has access.
Equitable access.
Equitable access. That's the key.
Yeah, I mean, these advancements should benefit everyone.
Everyone, not just a select few.
Who needs them?
Exactly.
I mean, another concern is just those unintended consequences.
Oh, always.
Right.
Yeah, because we're still learning about this complexity.
Yeah.
And what might seem like a simple fix could actually have ripple effects throughout the body.
Right.
We can't just think about things in isolation.
No.
We have to think about the system as a whole.
It's like an ecosystem, right?
It is. It is.
You tweak one thing over here, and who knows what's going to happen over there.
Exactly.
Yeah.
And, I think as we gain more and more control over our biology, it really raises these profound questions about what it means to be human.
It does.
If we can manipulate ourselves to live longer or to enhance our abilities, does that change our understanding of ourselves?
It's a big question.
It's a huge question. These are things that philosophers and ethicists have been thinking about for centuries.
Right.
And they're becoming more and more relevant as science advances.
We started this deep dive with a sense of wonder and possibility.
Yeah.
And I think it's important to hold on to that.
Absolutely.
But we also need to balance that with caution, right?
Yes. Caution and respect.
Yeah.
For the complexity of life.
Respect for life.
Yeah.
I couldn't agree more.
It's a delicate balance.
We need to continue unlocking these secrets.
Yeah.
But we need to do it responsibly.
Responsibly.
Ethically. With a clear understanding of the consequence.
Understanding the consequences, both good and bad.
Both good and bad.
Right. And it's not something that scientists can do on their own.
Right.
We need to have this open dialogue as a society to make sure these tools are used for the greater good.
This has been an incredible deep dive.
It has.
I mean, just mind-blowing discoveries and thought-provoking questions.
Yeah, it's really exciting.
And we've only just scratched the surface.
We've just begun to explore this.
Of what's possible.
Yeah.
But one thing is clear.
What's that?
The future of medicine is being rewritten.
It's being rewritten right now.
And these tiny proteins, these transcription factors, are playing a starring role.
They're the stars of the show.
Thanks for joining us.
Yeah, thanks for having me.
And, as always, keep exploring.
Keep exploring.
Keep questioning.
Keep questioning.
And keep learning.
Keep learning.